TGF beta signaling and stem cell dynamics in breast cancer growth. It is a great pleasure to introduce our speaker, Professor Mitsuyasu Kato. So Professor Mitsuyasu Kato is a young professor at the Faculty of Pharmacy Universitas Indonesia. We are very grateful to have him with us today. And he also is currently Executive Director and Provost of the University of Tsukuba. He previously also served as professor in the Department of Experimental Pathology, Faculty of Medicine, University of Tsukuba from 2002 to 2025. So he earned his medical degree and PhD in medicine from Tohoku University. His distinguished contributions have been recognized through several awards, including the Japanese Society of Pathology Academic Award, the University of Tsukuba Best Faculty Member Award, and the Japanese Pathology Award. So I used to call him Kato Sensei. Yeah, he was my supervisor during my PhD. So Kato Sensei, thank you very much for joining us. Shall we start? The virtual floor is yours.
Okay, thank you very much, Nerida. It's so excellent introduction. Shall I share my slides? Okay, so. Hmm. So, can you watch my slide?
Right, yes, Sensei, but still in the slide show. I mean the presenter mode.
Still, slideshow?
Yes.
Okay.
But yeah, it's presenter mode, so maybe
Presenter mode.
yeah, yes,
Okay,
yes.
wait. How about this?
Oh yes, all
Yes.
right.
Now, okay?
Okay, okay.
Okay, so let me start my lecture. Sorry for waiting. I'm so getting upset. I couldn't access the Zoom, but now, yeah, I'm very happy to talk with you. My lecture today is about TG-beta signaling and stem cell dynamics in breast cancer growth. So this is the content of my talk today. First, I will talk about general nature of cancer cells. And next, I talk about discovery of oncogenes and transforming growth factors. And third topic is the stimulus-inducing activity of cancer-promoting target genes and its related genes of transforming growth factor beta, PMEPL1, TMEPI, and GPLP. And fourth, I talk about stimulus induction promotes persistent increase of total cell numbers of cancer cells. I introduce a simulation model. And at the end, I want to talk about the remaining challenges in cancer research. So probably you know cancer is caused by sequence abnormalities and epigenetic modification of genomic DNA, which activates oncogene products and impair the function of tumor suppressor gene products. Cells with these abnormalities grow autonomously and excessively in multicellular organisms like let's see. Wait a moment. Sorry, I couldn't show the laser pointer. Mm, okay, anyway, I'll talk with that pointer. Anyway, cancer cells invade surrounding tissues and make distant metastasis. In this picture, this is the, let's say, vertical section of colon cancer, and cancer cells which originally developed in mucosal area, top surface tissue, invade and destroy muscle layers. Muscle layers now broken, and cancer cells spread over muscle layers. Okay, so how oncogenes discovered, I will talk in this slide. So first, researchers found normal cells and cancer cells give similar proliferation in ordinary monolayer culture conditions. In this case, normal cells doesn't mean cells directly separated from our body. These cases, people used establish normal cell clones. So these cells already get the immortality, so they can grow limitlessly in ordinary culture conditions. But in these conditions, even in these conditions, normal cells could not grow and make visible colonies on soft agar. But cancer cells did. So cancer cells can make proliferation even on soft agar and make visible colonies. By using this difference, Bishop and Balmas made interesting experiment. They infected tumor-forming virus to normal cells. Then normal cells also made colonies on soft agar, and this phenomenon was called viral transformation. So normal cells changed their phenotype and became the cancer-like growth pattern, made growth cancer-like growth pattern. So after this, this genes derived from virus was called viral transforming gene. And early age cancer researchers started to find which genes are responsible for this transformation. So, and found several viral transforming genes like SARC or RAS. After that, Delarco and Todaro found interesting phenomena. They just collected culture supernatant and added to normal cells. This NRK means normal rat kidney cells. Then normal cells without any genetic change, without transformation, they they made visible coordinates. So transform phenotype could be given by some Sobel factors. And many people started to find what kind of Sobel factor is, let's say, responsible for this. And 1981, Anita Roberts and Michael Spohn at NIH found for this function, two factors required. And one of them was transforming growth factor alpha. They gave the name, the factors, growth factors, which give transform phenotype. So and two factors required. Therefore, they called these activities as transforming growth factor alpha and beta. And those days, and other researchers already found epidermal growth factor, which is important for the regulation of normal cells, the regulation of the growth of normal cells, proliferation of normal cells. And transforming growth factor alpha turned out to be a family molecule of EGF, but transforming growth factor beta was totally new molecule. After that, people found many growth regulatory molecules, like growth factors, can be the origin of oncogene. Sometimes this origin of oncogene is called proto-oncogene, and by mutation, by multiplication or other abnormality, these genes can be active oncogene. And they found many of these oncogenes come from the growth factor itself or growth factor receptors, signaling molecules working downstream of growth factor receptors, and a transcriptional factor which works, which activated by growth signal. So growth factors and positive regulator of cell proliferation is one of the source of and major source of oncogenic modification of the genes. So now we understood TGF-alpha and other growth factors and their receptor or signaling molecules are the oncogenic genes. But for this colony formation, not only such genes, oncogenes, transforming growth factor beta also required. But why TG-beta also required, no one understood. Therefore, I wanted to elucidate why TG-beta required for this phenomena. And also, I thought not only in such in vitro experiment, even in our body, when cancer grow and make tumor, TG-beta signaling is activated. There are many evidences TG-beta is always activated in, let's say, pathological conditions in our body. So I thought cancer cells may commonly require cooperation of TG-beta to obtain immunogenic activity in our body. And I wanted to elucidate this hypothesis true or not. So after that, many researchers worked on TG-beta, how TG-beta binding to their receptors, signal, and change the cellular function. And in many cases, TG-beta changes the target gene expression, and it's required for the changes of cellular phenotypes. So anyway, if this is the major mode of function of TG-beta, oncogenic activity also mediated by some targeting method. Since TG-beta have so many different target genes and involved in so many different functions, I thought if we focus on one target gene, then we can get narrower function, and we can identify essential molecules important for tumorigenic function. So we worked on several molecules, and my talk today focus on PMEPA1 and GPND. So originally, this PMEPA1 was found to be TGBeta target genes, which expression is highly induced by TGBeta by Susumu Ito, who joined my lab in 2002. And he summarized his work on this molecule, which he did in Netherlands, and published this JBC paper. In this paper, he just mentioned PMEPA1 is highly used by TG-beta signaling. Those days, we have no information about the function of PMEPA1, but 2-3 papers already published. PMPP1 expression is highly activated in some cancer cells. So that's why we interested in this molecule and started to work on this. Soon after that, Caroline Hill in England also published one paper. This TMEPAI is the approved new name of TMEPA1 in 2005. And she found this TMEPAI is most highly induced by TG-beta signaling. And we continued working on this molecule and found this PMEPA1 is induced by TJ-beta. And this molecule binds TJ-beta signaling molecule SMAD. This PMP1 had SMAD interaction motif, and somehow suppress the TG-beta signaling. So PMPA1 works negative feedback regulator of TG-beta signaling. And we published our first paper on PMEPA1 in 2010. So it's already 7 years after we started working on this molecule. And at the same year, we published one more paper, how TG-beta signaling highly induced this gene expression. SMAD binding elements together with the transcriptional factor TCF7L2. This factor was famous regulator of Wnt signaling. So TG-beta signaling and Wnt-related transcriptional factor work together and induce this molecule. We also found epidermal growth factor, TGF-alpha type of signaling also activate this PMEP1 expression in the presence of TJ beta signaling. So this molecule is the unique has unique regulation. When TG beta and TGF alpha or EGF work together, then this molecule highly induced. Therefore, this molecule highly induced in cancer cells. So to analyze the function of this molecule, we started to make genomic modification, and we made targeted ablation of PMP1 gene and made the knockout cell line in parental cells. TJ beta stimulation induce PMP1 protein expression, but knockout cells totally lost the expression of these genes. And we could compare original cancer cell condition and PME, PA1 knockout condition. Then we can show the function of this induced PME, PA1 molecule. First, we compared ordinary cell proliferation in monolayer culture condition. Then this product did not affect so much on ordinary cell proliferation. But when we cultured without anchorage, sphere-forming protein condition, then in this condition, cell proliferation was highly suppressed by knocking out of PMEPA1. This is the in vivo tumor formation. In vivo tumor formation also depended on PMEPA1 expression. So this molecule is not required for cell proliferation, ordinary cell division, but highly required in vivo cancer growth or in vitro anchorage independent sphere forming growth, three-dimensional growth. So at this time, we didn't know what the difference between the cell proliferation in monolayer culture condition and sphere culture condition or in vivo condition. But now we found there is some difference. Okay, what the difference we wanted to know further? So for cell proliferation, two major signaling very important. One is RAS-MAP kinase pathway. It's important for the promotion of cell division. And the other let's say, signaling through PI3 kinase and AKT, important for cell survival and maintenance of the cellular life. So now we already know this molecule, PI3 kinase, is essential for the activation of this pathway. And the famous oncogenes, almost 30% of total human cancer have activating mutation in this PI3 kinase gene. And this PTEN is a negative regulator of PI3 kinase pathway. This is the phosphatase and get rid of phosphate from the target. And this AKT is the downstream regulator, a downstream kinase of PI3 kinase pathway. And in cancer cells, both threonine 308 and serine 473 phosphorylated and activate downstream pathway important for cell survival of cancer cells. But when people talk this story, people never talked about another molecule, PHLPP1. This is another negative regulator, phosphatase, which get rid of phosphate from serine 473. So even when PI3 kinase activated or PTEN inactivated, if PHLPP1 working, then serine 473 must be negatively regulated. But somehow in cancer cells, 2473 is also phosphorylated, but no one knew the mechanism. Therefore, we thought T-beta signaling through PMEPA1 negatively regulates PHLPP1 and activate AKT. So to elucidate this hypothesis, we made some experiment. For example, here we knock out PMEPA1, then PMEPA1 expression protein level in parental cells lost in knockout cells. In this condition, PMP1 in parental cells highly express PMP1, then suppress PHLPP1, so protein amount of PHLPP1 is very low. In this condition, AKT serine 473 highly phosphorylated. But when PMEPA1 knockout, then PHLPP1 protein level increase, then AKT serine 473 phosphorylation status reduced. So we also made one more experiment in PHLPP1, PMEPA1 knockout condition. We additionally knock down PHLPP1. So in parental cells, PHLPP1 is low, but highly activated in PMEPA1 knockout condition. But if we knock down PHLPP1 by siRNA, then PHLPP1 suppressed by this siRNA. In this condition, activated, so AKT phosphorylation highly activated in PHLPP1 knockout condition, and AKT phosphorylation, 3473 phosphorylation, reduced by knocking out of PMEPA1, but it recovered by knocking down of PHLPP1. By using such kind of experiment, we could show this left upper mechanism is working, and this molecular mechanism, cause the difference of sphere forming activities or colony forming activities. So we could show novel mechanism how PMEPA1 working through its double PY motif. It can bind to E3 ligase and regulate the protein degradation and regulate PHLPP1. and suppress AKT. So PMEPA1 can activate AKT. And we further elucidated this kind of mechanism at the end induce stem cell phenotype in cancer cells. Okay, so let make me switch to the next molecule. It's called glycoprotein NMB. This molecule we focused through another research history. First, we were working on the function of a transcription factor called MAFK, and we constitutively express MAFK in breast epithelial cells. This is immortalized normal cell line. Then again, it didn't affect on ordinary cell proliferation, but the sphere formation and in vivo tumor formation highly induced by MAFK. We could make some paper by this result, but this Okita-san, Yukari Okita was working on this project, and I suggested for her, okay, you found very interesting data. MAFK has highly tomogenic activity, but many people says transcription factor is difficult to target by drug. And you found one phenomenon important for cancer cell phenotype. But when we make excellent paper, we need to elucidate the mechanism, how it occurs. So now we found MAFK-induced tomogenic activity. How MAFK-induced tomogenic activity you need to work on, I suggested, and actually she did. Those days it was very tough work, but she found, she tried to show what kind of genes regulated by MAFK and found membrane protein, GPNB, is highly induced when she constitutively expressed MAFK in breast epithelial cells. So as a next step, we just expressed this target gene, GPNB, in NMUMG cells. We made multiple clones, and again, these new molecule didn't affect so much on monolayer culture growth, but highly increased 3D culture sphere forming growth. And when transplanted to mice, immunocomplemented mice, original cells, mock cells, made something mimicking normal breast tissue, but never make growing tumor. But only the expression of GPNB induced high tumor formation and easily made skin ulcer. This one gene expression induced cancer phenotype, constitutive proliferation and destroying surrounding tissue. So again, we found one more molecule very important for tumor formation. So next we used another breast cancer cells. It's mouse breast cancer cells 41, and we made 41 tumor in mice and collected tumor and separated into single cells, and we sorted cell surface GPNB expressing cells and raw cells by cell sorting system. Then, when we just checked, many cells had GPNB, but only 10% of cells had cell surface expression of GPNB. So we found GPNB is expressed only limited numbers of cells on cell surface. And these cell surface GPNB expressing cells, only these cells had stemness genes, SOX2 or nano. And it's only limited in the sphere. This is the visualization of surface expression of GPNB. And this green signals we get from inside of the cells, meaning GPNB is circulating cell surface and go into uptake and go into cells. This kind of GPNB positive cells has some essential role in cell proliferation in sphere culture condition or in vivo tumor forming condition. We sorted GPNB high and GPNB low cells, and we inject again to mice. Then GPNB high cells had 20-fold higher tumorigenic activity, meaning GPNB high cells. In the previous data I showed, GPNB high cells have stemness gene expression, and this is the stemness function. Tumor initiating function can be observed much higher in GPNB cell surface high cells. But some cells made tumor, even GPNB low cells shows some tumor, much lower incidence compared to GPNB high cells. So GPNB high cells had 20-fold higher frequency of tumor initiating function. So we analyze the tumor of made from GPNB low cells. Then we get the similar ratio of GPNB high and GPNB low cells in this tumor. So this GPNB high and GPNB low is not the different lineage. They can be transferred to the other condition. GPNB high cells can make GPNB low cells. GPNB low cells also make GPNB high cells in their conditions. Okay. So one more thing we focus is the difference of cellular kinetics in 2D and 3D condition. 2D condition, almost all cells Ki67 positive, meaning Ki67 is the marker molecule in cell proliferating cells, cell cycle running cells. So in 2D condition, monolayer condition, cells just continue to proliferate. But sphere condition, 3D condition, or in vivo tumor forming condition, cells proliferate, but they stop proliferation and die. Such kind of, let's say, multi-step life condition from proliferation and cell death recovered in 3D or in vivo tumor forming condition. That's the difference between 2D and 3D. And the surface GPNB expression, we could detect only Ki67 negative cells, meaning the surface expression of GPNB occur only when cells stop proliferation. Only growth arrested cells can make this phenomena. And this, GPNB high cells had 25-fold higher stem cell frequency. Okay, so we changed to the cultured condition of 41 cells to 3D condition. Then GPNB expression level highly induced. In this condition, stemless genes like SOX2, NANOG, CD44 also highly induced. So we next knock down GPNB. Knockdown efficiency not so high. GPNB was almost 50% or 60% reduced. Even this incomplete knockdown condition, stemless gene highly lost. That means GPNB expression and cell surface expression of GPNB protein essential for the stemness of cancer cells. So we made one more experiment. GPNB had very unique sequence, amino acid sequence in intracellular domain. This tyrosine XX leucine or isoleucine. This sequence is called item motif. And item usually have tandem repeat of this motif. So, it was very famous activating motif of immune receptors. This tyrosine is phosphorylated by SAC family and essential for the activation of immune cells. Therefore, we thought this tyrosine may be important signaling site and made amino acid change tyrosine to phenylalanine. So we made YF mutant of GPNB. Then when cells transfer to 3D condition, SOX2 and NARG stemless genes highly induced. But if GPNB is one amino acid change from tyrosine to phenylalanine, then stemless gene never induced. And in this condition, YF mutant cannot activate the sphere formation or tumor formation. That means this item motif or GPNB essential for sphere forming activity and tumor forming activity, and this is mediated by the phosphorylation of item tyrosine by SAC family. We published this paper, and ASCR, American Association for Cancer Research, found this is brand new study, and they made news in their Breaking Insights page of cancer research, and they said GPNB induced stemness in dormant breast cancer cells and introduced our work to the world. So now we found GPNB, this surface expression of GPNB rarely happen in cancer cells, and tyrosine phosphorylation by SAC family induce stemness. and this is essential for tumor formation. We also found DPNP has another function. This extracellular KLD domain essential for the activation of unit beta-catenin pathway. We also found this molecule interact to FGF receptor, and interestingly, in the presence of GPNB, AKT, which is important for cell survival, highly activated, but ARC, which is important for cell proliferation, reduced. So in the presence of GPNB, cell proliferation reduced, but cell survival or stemness induced. That brand new function we found. So this is the summary of the second part. Target gene of transforming growth factor beta, it's PMPA1, or some TJ beta related molecule, GPLB, commonly induces stemness in growth arrested cancer cells cooperatively with growth factor signaling and oncogene activation. That's what we found. Let's say why TJ beta required for tumor growth. So this is another story. In our tissue, we have multiple different regulations. And for example, this is colon mucosa. We have clit as a unit of proliferation, or let's say renewal unit. And we know proliferating cells located in the bottom. We can detect it by Ki67 expression, and we can detect DNA replication by incorporation of bromdeoxyuridine. This is the analog molecule of tyrosine. And we add BRD only 1 hour, then limited number of cells now in S phase of cell cycle. So among Ki67 positive area, we have several BRD positive cells. After that, we just waited 1 day, 2 days, 3 days, 4 days after addition of BRDU. Then BRDU positive cells originally located in the bottom gradually move up, and disappeared only just after 4-5 days. That means proliferating cells stop proliferation and move up and die from the surface. So such kind of cellular kinetics we can detect in tissue. So proliferating cells, tentatively increase cell numbers, but finally all cells die. But we can continuously have similar numbers of cells in our body. So for this mechanism, some cells we cannot see. Stem cells have essential role. Stem cells make asymmetric division and produce stem cells themselves and proliferating cells. Stem cells always give new proliferating cells. Therefore, we can keep some stable numbers of total cells in our body. So to detect stem cells in this tissue, we tried many antibodies, which is reported as stem cell marker, but everything failed. And finally, we started to check some, let's say, growth arrested or dormant cells in proliferative area. So in this experiment, we use osmotic mini pump, and this BRDU thymidine analog was continuously induced. Then 3 days, uh, during 3 days, proliferating cells become positive with BRDU antibody, and from the bottom, positive cells spread to the surface. But even in this positive area, we could detect limited number of BRDU negative cells. Even after 5 days, we could get BRDU, clear BRDU negative cells. That means these negative cells never had cell proliferation during the past 5 days. Okay. So after this experiment, we made serial section and reconstructed 3D structures and counted how many positive cells, how many negative cells we have in one clip. After such kind of analysis, we could make the, let's say, model how cell proliferation regulated in people. So for example, crypt of our intestine, we have stem cells, and it intermittently make asymmetric division to make same number of, to keep same number of stem cells. It's called self-renewal, and also make differentiated rapidly proliferating cells. They make serial division and stop proliferation and finally die. So if we program this mechanism is working, then total cell number always gets plateaued. How many numbers of levels, number of cells getting plateaued will be changed by how many times of serial division occur or how frequently stem cells make division. But anyway, it always becomes plateau. So when these left figure type of cell regulation is working, we always have plateaued number of total cell numbers. But even in, let's say, abnormal condition, in this condition, tumor suppressor genes is APC is mutated, and we have some small bad sprout of cancer we have. But these abnormal condition never make visible tumor. And when we add some drug called DSS, this induces inflammation into the colon. Then tumor formation induced. So by adding DSS, we can change silent small tumor to visible tumor. And this occurs only in the presence of both mutation and DSS addition, and without only one, for example, DSS treatment never give tumor formation to wild type mice. So both require. So before DSS treatment, tumor cells, adenoma cells keep silent in small clusters. But after inflammation induction, they start to make visible tumor. And in this silent small tumor condition, we have proliferating cells, but the number of proliferating cells are very low frequency. And when we add BRDU, then most cells become positive after 7 days. That means these growth arrested cells at least divide once in 7 days. Okay? So such kind of regulation is working, and such kind of evidences tell us we have slowly or intermittently proliferating stem cells. And we also made one more hypothesis. These growth arrested cells can induce stemness by the function of GPNB or PMEPA1. Then cells can return from this condition to this condition. But if we program like this, then yeah. So okay, so in this model, cells keep small numbers in early time point. But once cell clusters start to grow, then continuously grow the total cell number. Such kind of unique kinetics was discovered. So cells keep plateaued number, but once stem cells start to grow, then total cell number start to grow. And I thought this is this is the mechanism of cancer-specific autonomous and monogenic cell growth. And GPNB and PMPA1 is essential molecule for this phenomenon. Therefore, if we target GPNB or PMPA1, we can make a brand new method to regulate cancer cell proliferation. Okay, stem cell induction breaks dynamic balance of total cell numbers and induces cancer-specific persistent increase of total cell numbers. So now, even the most famous pathology textbook, cancer is explained by the word hallmark of cancer. Hallmark of cancer is something like cell sufficiency, growth signal, insensitivity to growth inhibitory signal, something like that. Cells cannot die. Cells lost aging related stop of proliferation. If it's true, normal cells can make plateaued total cell number, but cancer cells never stop cell division and always increase. Is this correct? Probably in monolayer cell culture condition, this kind of regulation occur, but it must be different in tumor forming or sphere culture condition as I talked today. So now we know stem cells are important for self-renewal, tumor initiation, and relapse and metastasis. And why? But if we don't understand the, let's say, limited number of stem cells, only stem cells can make tumor. We don't understand why non-stem cancer cells cannot initiate tumor formation if cancer cells always divide and never die. So now I hope you understood from my talk, even cancer cells can stop proliferation and die. Therefore, they make stable numbers for a long time, like invisible tumor cell clusters in mice intestine I showed today. But sometimes growth-arrested cancer cells can induce stemness. If so, stem cell numbers increase and the plateaued total cell number increase from the first condition. And if total cell number increase, ratio of stem cell induction increase. So cells gradually accelerate the increase of total cell number. And for this hypothesis, we already have multiple evidences stem cells induced in cancer cell clusters, but we don't have any evidence. So for this, these almost dying cells must get new lifespan. Renewal of lifespan is essential when non-stem cancer cells return to stem cells. And this is the, let's say, questions still remain. We need to work on, and Noriza actually had many interesting data on this. I just introduced one, let's say, slide she made. Asymmetric cell division is not so much studied in cancer cells, but many people working on this asymmetric division in fission yeast. And in fission yeast, there are some barrier between mother cells and daughter cells. And for example, some aging-related molecules can be kept only in mother cells and never transferred to daughter cells. Therefore, daughter cells get new life. Okay, and full length of cell division life. Such kind of mechanism may be working even in cancer cells. That's what we need to elucidate in the future. Okay, I think this is the end of my talk today. So if you have any question, I'm happy to answer to your question. Okay,
Thank
Yurita, can you
you.
hear me?
Yes. Thank you very much for the insightful and engaging talks on the TGF beta signaling and stem cell dynamics. So from these talks, we have now get more clearer about the view of how TGF beta signaling and stem cell dynamics can shape the breast cancer growth and progression. And after this, we will now continue with the discussions and question and answer session. So for the participants who would like to ask a question directly, may use the raise hand feature, and you can ask directly to Professor Kato. So anyone uh, would like to ask question? I think also we have a question from the chat box. So from Anissa Kisti Masrio, uh, thank you very much for the wonderful lecture, Professor Mitsuyasu Kato. I have questions regarding the slide before the GPNMB section. So what does it mean when the result shows that monolayer growth remains unchanged, but there is an extreme change in the sphere formation?
Okay, so in monolayer culture condition, it's very different from in vivo condition or sphere culture condition. First, usually we have serum, and probably normal process, proliferating cells stop proliferation, differentiate, and die. Such kind of cycles are probably blocked by this special condition. So cells, as I showed in the end, cancer cells continuously divide and increase total cell number. Such kind of cell proliferation occur in monolayer culture condition. But it's not the case in vivo. In vivo, even cancer cells can stop proliferation and die. But total cell number continuously increase by other mechanism. Today I talked stem cell induction. Such kind of stemness-dependent cell proliferation required for sphere formation or tumor formation. So sphere forming condition, when we talk about the lifespan, so Hayflick limit is the most let's say famous, and it's caused by shortening of the telomere, but it's much longer aging process. Even in normal epithelial cells or hematopoietic
Sorry,
cells,
anak
cell
aku penelitian. Boleh lihat list nama pasien yang konsul ke dokter.
so cell proliferation stops much earlier than Hayflick limit. So such kind of mechanism working even in cancer cells, and it makes difference in the growth of monolayer culture condition and spare or tumor forming condition. So I hope you understand.
Thank you, Professor Kato, for the explanation. So I hope this will answer, the questions from Anissa Kisti. Okay, anyone would like to ask questions? So maybe while waiting the others to ask questions, I have a question. So,
You know much
recently,
more than me about this.
so it's very interesting that recently Professor Hanahan update about the hallmark of cancer to expands the list of the hallmark of cancer, expanding the framework beyond the cancer cell intrinsic mechanism. So how do you think the position of this TGF beta signaling in this new update from the Professor Hanahan?
Okay, so TG beta has so many different functions, and TG beta has many roles in regulation of cell proliferation, cell differentiation, cell-cell contact, matrix formation. So it's difficult to say TG beta is everywhere. TG beta is working everywhere to make multicellular organisms. So cancer is the disease with abnormality in regulating cell proliferation. So of course, TG beta has some role in this. So anyway, sometimes TG beta signalings also work as tumorigenic or tumor suppressive signaling pathway to stop proliferation. But when cells get resistance to growth arrested function of TG beta, then another function of TG beta promoting tumor formation or cell proliferation get up, I would say, become the majority of function of TG beta. So anyway, when we talk about TG beta, everyone says it's so complicated and difficult to understand. I also, even after I'm working on TG beta more than 40 years, but still I feel TG beta is difficult to understand. But yeah, in cancer cells, multiple function, of course, TG beta has. But this stemness, TG beta has essential role to induce stemness or to stop proliferation. So growth arrest is essential for stemness induction. Such kind of, let's say, kinetics. We have to think about kinetics, not status. That's very important to understand cancer. So, sorry, I don't have clear answer, but probably you need to work on. Okay,
Yes, yes.
is it okay?
It's actually the following questions is related to the TGF-beta signaling that very complex, especially
Yeah.
how the it's involving cancer progression or something like that. So
Mm.
in your opinion, what advice that would you give to us for the young researcher or students who are just starting the research in this field or the other fields? Like, what kind of advice would you give to us?
Okay, so it's something like research topics we encounter unexpectedly, but after that you can select which next experiment you select. So and you get result and think next experiment and get result and select which direction you go next. So don't think so much, seriously what you are doing now. 1 year later, 2 years later, 10 years later, probably your path you walked become very, original. So anyway, continue working on selection of your future direction. Then it will make you, let's say, only one researcher in your field. Is this okay?
Very, nice advice. Okay. I will check if a participant would like to ask question. I think because the time is nearly end, so I would like to thank Professor Kato for very insightful talk and taking the time to also answer the questions from us. So we also sincerely thank all participants also who join or participate in this guest lecture. So before we conclude, we would like to ask for your kind assistance in completing the attendance form. So the organizing committee already provided the link for the attendance. So please fill out the form. And we also would like to take a picture together. So please everyone to turn on your cameras, and we would be delighted if as many participants as possible could join us on camera. Maybe Mbak Alia, please help us to take the picture.
Hello, Prof. Kato.
Hi, hi.
Hi, nice to meet you again. Yeah, thank you for this lecture.
Thank you. Yeah.
Kata Sensei, could you please help us to slideshow? I mean, close the presentation.
So, wait.
All right, Prof. Kato, Bu Nuriza, and all participants. I would like to take the picture together for first slide. First slide first. Okay, please open the camera. Okay. Seleksi calon. Okay, 3, 2, 1. And the second slide. Okay, 3, 2, 1. There are 5 slides, but I will take the last for the third slide. Okay, 3, 2, 1. Okay, thank you very much. Okay, thank you so much
So
for
this time I'm very sorry I could not come to Indonesia, but I try to find my time to visit.
Yeah.
So actually, our university staff go to UE in September, I heard. September 2nd and 3rd, probably they have some university fair and visit UE.
Yeah.
So
I heard about that.
you have? Okay.
Together with Chiba University, right?
Yeah. At
Yes.
least Kyosuke Nagata, our president, will come to UE. So
Will you join also?
they didn't ask me.
Uh,
I thought I should go, but not involved in the member.
yeah, I hope next time you can
Yeah,
visit
yeah.
us. Yes.
Yes.
Yeah. Okay. So once again, thank you so much, Professor Kato, for